Welcome to the Deep Dive. We're the show that tackles complex science, breaks it down, and really gives you the key to understanding the latest insights shaping our world.
That's the goal.
Today, we're plunging deep, right into a microscopic battlefield inside your own body. Seriously, imagine your immune system, your ultimate defense force, programmed to fight off invaders. But suddenly, it encounters an enemy that looks, well, disturbingly similar to parts of you.
A case of mistaken identity, essentially.
Exactly. We're focusing on a tiny but incredibly powerful piece of viruses, the receptor binding domain, or RBD. And this isn't just about how viruses sneak into cells.
No, that goes deeper.
It's about how these minuscule viral keys can actually rewrite your immune system's instructions, sometimes with, well, pretty devastating results.
That's right. So in this deep dive, we're going to journey from the nuts and bolts. You know, how viruses like SARS-CoV-2 use their RBD to latch onto specific hosts, through how our immune system learns to spot these viral signatures. But the real crux, a really critical part, is exploring these things called preserved epitopes.
Preserved epitopes, right.
These are specific recognition points on the RBD. And the unsettling thing is how they can sort of trick the immune system.
Trick it how?
Well, instead of clearing the virus effectively, the immune response might get shifted. It might help the virus evade detection or, in the worst cases, turn against the body itself, sparking autoimmunity.
Wow, okay. So our mission for you, listening in, is to give you that shortcut. We want you to really get this cutting-edge science. You should walk away with some profound insights into, how viruses pick their hosts, the details of immune recognition, and this really complex, sometimes alarming dance of autoimmunity.
By the end, you'll understand these biological mechanisms, sure, but also how they impact our health, how we respond to global challenges, even how we design future defenses.
Yeah, get ready for some surprising facts. There'll definitely be some aha moments, I think.
I expect so.
Okay, let's kick off with the basics. This receptor binding domain, the RBD, fundamentally, what is it?
Think of it like the very specific tip of a key the virus uses to unlock and get inside our cells.
And for SARS-CoV-2, this RBD isn't just floating around, right? It's part of that bigger structure.
Exactly. It's a crucial projecting piece of its larger spike protein, the one that gives the coronavirus its crown-like look. The RBD is right out there on the surface.
Got it. And you mentioned the spike protein isn't just a fixed shape.
Not at all. That's a key point. It's incredibly dynamic. It exists in different 3D shapes or conformations. Think closed, intermediate, and open forms.
Like a tool that can adjust itself.
Precisely. Not a rigid key, more like a sophisticated multi-tool. This flexibility is absolutely essential for its job. It lets the virus kind of optimize its approach to binding our cells.
And there's another step needed, something about cleavage.
Yes, furin cleavage. To be fully active, to really enable entry into the cell, the spike protein needs this specific molecular SNP.
Furin cleavage. Help us visualize that. What's being snipped?
Okay, imagine the spike protein has a sort of molecular safety catch on it.
Right.
Until that catch is precisely cut off by an enzyme in our own cells called furin, the viral key can't fully extend or adopt the exact shape it needs to unlock the cell door properly.
So furin is like the pair of molecular scissors inside us that the virus exploits.
You could put it that way, yes. It performs that crucial SNP, effectively activating the spike protein, making it ready for infection. And what's amazing is the level of detail we now have.
From structural mapping.
Exactly. Using advanced techniques, scientists have generated these best quality maps, especially for the uncleaved spike protein in its closed state. This lets them model the entire protein chain almost atom by atom.
Wow. That level of detail must be critical.
It's foundational. It underpins everything we understand about how the virus interacts with us and, crucially, how we might be able to stop it.
Okay, so the RBD is the key tip. Furin cleavage helps activate it. How does this active key find its specific lock, the host cell receptor?
For SARS-CoV-2, the main lock is a receptor called ACE2. You've probably heard of it. Often another enzyme called TMPRS2 helps out too, facilitating entry after the binding happens.
And this lock and key interaction, ACE2 and the RBD, that's what determines host affinity.
That's the core of it, yes. Host affinity, which species of virus can infect, and even which cell types within that species. It's incredibly specific. It's the difference between a virus being, just a bat virus and one causing a human pandemic.
The molecular details must be pretty intricate there.
Oh, absolutely. The interface where the SARS-CoV-2 RBD touches the ACE2 receptor isn't just a flat contact. It involves several additional stabilizing interactions.
What kind of interactions?
Think weaker molecular forces, things like hydrogen bonds, electrostatic attractions, van der Waals forces. They're subtle individually, but together they create this really snug, stable fit. It ensures the key turns the lock efficiently.
And you can see the difference compared to related viruses, like bat viruses.
Definitely. Take bat RataG13, it's genetically very close to SARS-CoV-2, but just a few differences in key amino acids in its RBD.
Like tiny changes on the key's teeth.
Exactly. For instance, RataG13 has histidines at positions 505 and 440, where SARS-CoV-2 has a different residues. These small changes might lead to what we call potential steric repulsion.
Steric repulsion.
Yeah.
A molecular clash, like the key bumping against something in the lock.
That's a great analogy. It's a physical clash between the atoms that prevents that perfect snug fit. So for RataG13, the key doesn't fit the human ACE2 lock quite as well.
And that affects its ability to infect human cells.
Significantly. It makes it much less efficient. And this raises a massive question for scientists trying to predict the next pandemic. What tiny, almost invisible changes in an animal virus's RBD are needed for it to jump to humans?
It's mind-boggling. A single amino acid swap could be the difference between a virus staying in bats and sparking a global crisis.
It's evolutionary fine-tuning at an astonishing level. Those tiny changes in the key's teeth deciding if it opens a human door or a bat door. It's incredible.
But scientists aren't just watching this happen, right? They're actively using this knowledge, especially for vaccines.
Indeed. There's a huge effort focused on manipulating these RBDs, specifically creating highly refined recombinant RBD proteins in the lab.
Recombinant meaning engineered pieces, not the whole virus.
Exactly. They're produced, usually in cell cultures, purified, and their structure is meticulously checked. Then comes the crucial testing for cross-reactivity.
Cross-reactivity, seeing if antibodies against one virus work against another.
Precisely. Can antibodies raised against, say, the RBD from the original SARS virus, SARS-CoV-1, also neutralize SARS-CoV-2?
And can they?
Some early data was quite promising, suggesting yes, those SARS-CoV-1 antibodies can offer some cross-neutralization against SARS-CoV-2. Points to shared vulnerabilities.
But not all coronaviruses.
Right. Contrast that with MERS-CoV. Antibodies generated against the MERS RBD seem to only neutralize MERS itself, not SARS-CoV-1 or SARS-CoV-2.
Interesting. So that tells us about how closely related they are structurally.
Yes, it guides our search for common targets, for potentially broad-spectrum vaccines or treatments. It highlights the structural differences and similarities.
So how is this knowledge shaping actual vaccine design now? It sounds like we're getting much more sophisticated than just showing the immune system a weakened virus.
We really are. The cutting edge involves nanoparticle-carried subunit vaccines. The basic idea is to move beyond just injecting the viral protein, like the RBD, by itself.
Why? Doesn't that work?
It can, but presenting it on a nanoparticle often works much better. Think of these nanoparticles as miniature, highly organized display stands.
Okay, like showcasing the important part.
Exactly. Platforms like ferritin, which naturally forms a sphere made of 24 identical protein units, or larger 60-unit spheres like E2P and I301V9 are used. They can even be engineered with extra bits to boost the immune response, like T-cell epitopes.
T-cell epitopes.
Oh.
To get T-cells involved too, not just antibodies.
Precisely, like adding PDRE sequences. But a really critical design choice is the linker, the molecular tether used to attach the RBD to this nanoparticle scaffold.
The linker length matters. How?
Oh, it matters a lot. It influences how densely the RBD molecules pack onto the nanoparticle surface and, this is key, the accessibility of unfavorable epitopes on the RBD core.
Unfavorable epitopes. Okay, that sounds bad. What makes an epitope unfavorable?
Good question. An unfavorable epitope is a part of the RBD that, if the immune system targets it heavily, might lead to antibodies that aren't very protective. Or worse, antibodies that could potentially cause harm or distract the immune system from the real targets needed for neutralization.
So you want to hide those parts.
Exactly. The goal is to minimize their exposure. You can do this by optimizing that linker length using short, flexible sequences like GS-GSG, perhaps repeated, which encourages the RBDs to pack in a way that buries those unfavorable bits.
However, any other tricks?
Yes, masking them with glycans sugar molecules. The MERS-CoV RBD, for instance, naturally has 3 spots where these sugars attach. You can engineer these to act like molecular camouflage, hiding those unwanted regions.
So the hypothesis is less access to bad epitopes means a better immune response.
That's the driving idea. Nanoparticles designed this way should have enhanced efficacy because the immune system is guided to focus its firepower on the protective epitopes. This detailed structural design is all aimed at generating broadly neutralizing antibodies.
The Holy Grail, right. Antibodies that work against many variants.
That's the ultimate goal, protection against not just one strain, but potentially multiple current and future variants.
Okay, let's switch gears slightly and talk about these epitopes themselves. Our immune system is incredibly specific, you said. It doesn't see the whole virus.
No, it zooms in. It recognizes tiny, distinct molecular shapes or sequences on the pathogen. Those are the epitopes. They're the antigenic determinants.
Like recognizing a specific feature, not the whole person.
That's a good analogy. It's like a security guard focusing on a unique scar or a specific tattoo, not just the overall appearance.
And there are different types, B-cell and T-cell epitopes.
Correct. B-cell epitopes are usually 3D shapes on the surface of the antigen that antibodies can bind to directly. T-cell epitopes are typically short, linear peptide fragments that T-cells recognize, but only when they're presented by other specialized molecules called MHC molecules.
MHC, major histocompatibility complex.
Yes. So B-cells see the target directly. T-cells need it presented to them on an MHC platter, so to speak.
So like the security guard recognizing someone in a lineup directly, T-cell, versus needing another officer, MHC, to show them a specific photo, T-cell epitope.
Excellent analogy. That captures the difference quite well.
How do scientists actually find these tiny targets on something complex like SARS-CoV-2?
It involves a lot of sophisticated computational work. They assign structural properties to a protein using huge databases like the Protein Data Bank, the PDB, which stores known protein structures.
So they compare the virus proteins to known structures.
Partly, yes. And they use tools like Scratch to predict properties like which parts are likely to be on the surface or what their local structure might be just from the amino acid sequence. They've done this for the entire SARS-CoV-2 proteome, all its proteins.
Using techniques like threading, what's that?
Threading is like taking the virus protein sequence and trying to fit it or thread it onto known 3D protein folds from the PDB database. It helps predict the overall shape.
And ab initio side chain prediction.
That's trying to predict how the individual amino acid side chains the bits that make each amino acid unique will arrange themselves in 3D space based on fundamental physics, building it from the beginning, ab initio.
So it's like reverse engineering the virus's structure to find its weak points, its recognizable features.
Precisely. And a key goal here is identifying potentially cross-reactive epitopes, not just within SARS-CoV-2, but epitopes shared between the virus and human proteins.
The mimicry angle again. We'll definitely come back to that.
Yes, it's crucial. The amount of data is staggering. Analyzing thousands of potential epitope windows required developing special interactive web applications just to navigate and make sense of it all.
Okay, so we map all these potential epitopes. Why is it so important to know which ones are immunodominant? Doesn't the immune system just attack all of them?
Not equally. Immunodominance means some epitopes trigger a much, much stronger immune response than others. The immune system focuses its attention.
That sounds good, right? Focus the attack.
It can be good, but it's a double-edged sword. If the immunodominant epitope isn't actually very good at stimulating protective immunity.
Or if it's one of those unfavorable ones.
Exactly. Then the immune system might waste a lot of energy on a less effective target, or even a harmful one, while neglecting more critical vulnerabilities.
So the immune system could be focusing on the wrong thing.
It happens. That's why predicting which epitopes will be immunodominant and how well they bind to MHC molecules is so important. They use tools like an amino acid similarity matrix for peptide, MHC binding, to help predict this.
A compatibility chart for epitopes and immune presentation molecules.
Sort of, yes. It helps predict how likely a given T-cell epitope is to be presented effectively, which is crucial for initiating a T-cell response.
So if the immune system might focus on the wrong targets, can we redirect it?
That's a major goal of advanced vaccine design. Refocusing neutralizing antibody responses by targeted dampening of an immunodominant epitope.
Dampening, like telling the immune system to ignore a specific part.
In a way, yes. Or making that part less visible. There was interesting work with HIV's GP140 protein. By chemically cross-linking it, stabilizing its shape.
Locking it in place.
Right. They could enhance the neutralizing antibody response by guiding the immune system towards more critical protective sites and away from distracting non-neutralizing ones.
And this applies to SARS-CoV-2 vaccines, targeting specific populations.
Yes. The goal with modern SARS-CoV-2 vaccine formulations, especially those considering MHC class 1 and 2 epitopes, is to account for human haplotype distributions.
Haplotypes, the genetic variations in MHC-HLA molecules between people.
Exactly. Different people present peptides differently. By understanding these distributions, we can try to engineer vaccines with epitopes that are likely to be presented effectively across a broad range of human genetic backgrounds.
So trying to make the vaccine work well for almost everyone, despite our genetic diversity.
That's the ambition, engineering the vaccine to guide the immune response effectively, rather than just letting the virus dictate where the immune system focuses.
Okay, now let's take that turn you flagged earlier, the really critical, maybe unsettling part.
Yeah.
What happens when a viral epitope, a target for our immune system, looks almost identical to a protein in our own body?
That's molecular mimicry, the formal definition. Cross-reactivity against shared immune epitopes of proteins of human and invading pathogens.
The enemy looks like family.
Precisely. The immune system, trying to fight the virus, sees this look-alike sequence on one of our own proteins and mistakenly attacks it.
And this isn't just theoretical for COVID-19.
Sadly, no. It's strongly suggested as one of the mechanisms that could initiate dysfunctional immune responses leading to autoimmune and inflammatory diseases following SARS-CoV-2 infections.
So the body fighting the virus accidentally starts fighting itself. That's genuinely terrifying. What's the evidence?
There's a concerning increasing number of reports of autoimmune and inflammatory diseases popping up after COVID-19. Things like MISC, that's severe inflammatory syndrome in kids.
Multisystem inflammatory syndrome in children.
Right. And also findings from kidney biopsies in COVID patients. They sometimes show autoimmune-like damage, immune complex deposits that aren't directly caused by virus in the kidney itself. It suggests the immune system has gone rogue post-infection.
Can you give us some concrete examples? Where are these molecular lookalikes found?
Yes, research has pinpointed some specific troubling examples. One segment of the SARS-CoV-2 spike, residues 712, 720. The sequence is IAIPT-NFTI. It shows similarity to a human protein called SLC5A6, which transports vital nutrients. The human sequence is FSLPTNLTV, close enough to potentially confuse the immune system.
And that's linked to specific diseases.
It's been associated with psoriasis, especially in people with the HLA-C12.03 gene variant, and also ulcerative colitis with HLA-C12.02. Those HLA types might be better at presenting this mimicked peptide.
Wow, another example.
Another is spike protein residues, 446460, GGNY-LRRRLFRKKN, an enzyme really important for adding sugars to proteins, especially in muscle and brain.
And the consequence of attacking that.
This mimicry is linked to serious conditions like multiple sclerosis and systemic lupus erythematosus, particularly in folks with the HLA-DRB108.01 variant.
It's incredible how specific these links are. Any others?
Yes, there are more. Potential mimicry involving respiratory pacemaker neurons possibly linked to Guillain-Barré syndrome via similarity to human heat shock proteins. Also, human molecular chaperones proteins that help other proteins fold share epitopes with SARS-CoV-2. Attacking these could lead to autoimmunity against the lining of our blood vessels, endothelial cells.
Which could explain some of the clotting issues seen in severe COVID.
It's certainly a plausible contributing factor, and we see similar HLA associations in other autoimmune diseases like Crohn's or Graves' disease, further illustrating this general principle of genetic susceptibility interacting with triggers like mimicry.
So it's not random. It's a specific targeted mistake by the immune system based on these shared sequences. A precise case of mistaken identity.
Exactly. And the cells involved are these autoreactive T cells and B cells that somehow slip through the net during immune system development.
They escaped the quality control.
They escaped central tolerance in the thymus and bone marrow. Normally, these escapees are kept quiet by peripheral tolerance mechanisms out in the body.
But infection can wake them up.
Under certain conditions, yes, especially strong inflammation. These dormant, self-reactive cells, sometimes called sleeper B cells, can get activated.
You mentioned bystander activation, too.
Right. It's not just the direct mimicry. The intense inflammation during an infection creates a chaotic environment. Inflammatory signals, cytokines are everywhere.
Like alarm bells ringing constantly.
Exactly. And these signals can activate nearby T cells and B cells, even if they aren't specifically recognizing the virus or the mimicked self-peptide. They just get caught up in the general alarm. So you have mimicry and bystander activation potentially working together.
A perfect storm for autoimmunity.
It really is. A double hit that can push the system over the edge.
And when the system goes this haywire, that's when we hear about the cytokine storm.
Precisely. The cytokine storm is that uncontrolled activation of inflammation. It's a hyperinflammatory state that becomes self-perpetuating.
A deadly loop.
Yes, the sources describe it as a loop of pathological immunity, pathological inflammation, pathological thrombosis. Basically, misdirected immune attack, tissue-damaging inflammation, and excessive blood clotting.
Leading to.
Tissue damage, microcirculation blockade, and severe multi-organ dysfunction. The body's response becomes the main source of harm. Tracking levels of cytokines like IL-6 can actually help predict how severe these events might become in COVID patients.
Are there specific immune cells driving this storm?
Key players include Th17 cells and the cytokines they're associated with, particularly interleukin-6, IL-6, and interleukin-17, IL-17.
How do they interact?
IL-6 is a powerful signal. It pushes naive CD4 plus T cells to differentiate into these Th17 cells. It does this via a specific signaling pathway inside the cell involving something called STAT3 activation.
So IL-6 flips the switch for T cells to become Th17.
Essentially, yes. And these Th17 cells are known to be major drivers in many autoimmune diseases: psoriasis, psoriatic arthritis, ankylosing spondylitis. They're also implicated in multiple sclerosis and inflammatory bowel disease.
So they're often involved when inflammation causes tissue damage.
Yes, they're found in these pathological situations driving tissue destruction and autoimmunity. And the combination, the synergy between IL-6 and IL-17 seems particularly linked to the severity of COVID-19. They're really fanning the flames of that cytokine storm.
So the very response meant to clear the virus creates the conditions for autoimmunity.
Right.
It fundamentally switches the immune system's focus.
Exactly. And this brings us right back to that core idea, how these preserved epitopes could switch immune response from virus clearance to immune evasion.
How does that switch happen? If the immune system is busy attacking self-antigens?
Its ability to effectively find and eliminate the actual virus can be seriously hampered. Its resources are misdirected, it's distracted, or the self-inflicted damage weakens the overall host defense.
So instead of clearing the invader, the immune system is harming the host and maybe inadvertently helping the virus hang around longer or cause more trouble. The ultimate betrayal.
It becomes truly dysfunctional. The system might be practically inactivated against the virus, or its over-the-top reaction directly contributes to the pathogenesis of severe COVID-19 symptoms, like the organ damage. It's like in cancer, where tumors evolve ways to escape immune attack. Here, the virus, or the autoimmune chaos it triggers, allows it to evade effective control.
So the mechanisms designed for defense, through mimicry and inflammation, lead to self-attack or just ineffective viral control. That's profound.
It's a profound shift in immune priorities, with potentially devastating outcomes.
Okay, understanding this molecular mimicry, this potential for self-attack, must make vaccine development incredibly tricky. How do you design a vaccine that's strong against the virus, but doesn't accidentally trigger autoimmunity? It's like walking a razor's edge.
It absolutely is. It requires extreme care. You need meticulous sequence analysis of observed HLA-30 study peptides from the virus.
Making sure the vaccine targets are unique to the virus.
Exactly. Ensuring minimal overlap with SARS-CoV, and crucially, no match with other CoV spike proteins or any human sequences. Finding that unique viral fingerprint that won't be mistaken for self.
And we already see some immune shifts after vaccination.
Some studies show subtle changes. For instance, in children, COVID-19 vaccinations were observed to alter cytokine responses to heterologous stimulants, meaning their immune cells responded differently to various non-COVID triggers a month later. Specifically, increased responses involving innate, inflammatory, and notably, Th17 markers were seen when stimulated with viral components.
So the vaccine can subtly retune the immune system more broadly.
It suggests it can, yes, highlighting the need to really understand these downstream effects, especially regarding inflammation and potential autoimmunity pathways.
We've heard discussions about potential side effects, like muscle or joint issues after vaccination. What did the sources say?
They acknowledge these reports. There's an increasing number of reports of autoimmune and inflammatory diseases following SARS-CoV-2 infections, and the sources stress that studies are needed to figure out the mechanisms that link COVID-19 vaccines to inflammatory musculoskeletal disorders.
Are there specific examples?
One study did note that shoulder-related disorders like rotator cuff problems or frozen shoulder were notably higher after COVID-19 vaccination compared to other issues, almost double the expected rate. That specific pattern needs more investigation.
And you mentioned a harvesting effect. That sounded complex.
It is, and it relates to interpreting population-level data on adverse events. The idea is, if a vaccine has rare but serious immediate side effects, these might disproportionately affect people who are already very frail or vulnerable.
So those individuals might have a negative outcome early on.
Potentially, yes.
Right.
They might be harvested from the population earlier, so to speak. This means if you only look at data from later doses or further out in time, you might miss the effect because the most susceptible group isn't fully represented anymore.
So one study found a higher risk of death after early doses.
One study did report a higher risk of all-cause death after 1 or 2 doses compared to the unvaccinated, suggesting that while COVID deaths might decrease, this could be offset by increases in other causes of death, possibly linked to direct or indirect vaccine effects in vulnerable subgroups.
That doesn't negate the overall benefit for most people, but
And it absolutely underscores the need for very careful long-term follow-up, detailed analysis, breaking down data by risk groups, and transparency in investigating these potential mechanisms. We need the full picture.
this whole discussion about precisely targeting immune responses and the potential for misdirection also brings up a much darker possibility: biological weapons.
Unfortunately, yes. The technology to create lethal new strains of viruses and bacteria is becoming more accessible. Pathogens can potentially be manipulated to be more stable, harder to detect, or to evade current defenses.
Like combining viruses. Chimeras?
Concerns have been raised, for instance, by former Soviet bioweapons scientists about creating chimeras, maybe blending features of Ebola and smallpox. And the relevance here is terrifying. If someone intentionally engineered preserved epitopes into a bioweapon.
Epitopes designed to mimic human proteins.
Exactly. They could weaponize molecular mimicry, create a pathogen that not only infects but actively triggers autoimmunity or disables the immune response against itself. It's a chilling prospect.
Which brings us back to that phrase, horror autotoxicus. What did that refer to again?
Horror autotoxicus, literally the horror of self-toxicity. It was coined by Paul Ehrlich, a founder of immunology, to describe the body's intrinsic aversion to attacking itself. But it's also used to describe when that fails catastrophically.
Well, like in that mouse experiment.
Yes, the source mentions scientists studying myelin peptides who accidentally triggered a mechanism that caused mice to self-destruct in a condition known as horror autotoxicus. The immune system attacked the nerve sheath with devastating results.
A stark warning.
It's a powerful illustration of what happens when immune recognition goes profoundly wrong, when the defense system turns inward with lethal force. A real cautionary tale from biology.
But looking towards solutions, this detailed knowledge must also guide personalized medicine, right?
Absolutely. Understanding individual genetic variation, especially in our HLA genes, the MATZ molecules, is critical. These variations dictate so much about our susceptibility to autoimmune diseases and how we respond to infections and vaccines.
That's the field of immunogenetics.
Yes, mapping the immunogenetic architecture of autoimmune disease. Trying to understand precisely how our genes predispose us. It's complex, though. Genome-wide studies, GWS, have limitations. Sometimes effects are underreported or masked by other nearby variants. We need really detailed, fine mapping studies.
To pinpoint the exact genetic factors influencing individual immune responses.
Precisely. The more we understand these molecular interactions in our individual genetic blueprints, the more we can tailor prevention and treatment, moving away from one size fits all.
So we've really journeyed deep today. From the viral RDD key to how our immune system spots epitopes, and then into this really unsettling territory of molecular mimicry.
Microscopic details with potentially huge consequences for our health.
And what stands out is how one feature, like a conserved epitope, can be this pivot point.
Yeah, it's fascinating and frankly a bit sobering how it can potentially flip the switch, moving the immune response away from clearing the virus towards evasion or even self-attack. It just highlights the incredible complexity, the built-in vulnerabilities, and the delicate balance of our own biology.
Our immune system. Powerful, precise, but also potentially tricked by these molecular lookalikes.
It's exquisitely evolved but not infallible, susceptible to misdirection.
So what's the big takeaway for us? It reveals huge challenges, but also opportunities in immunology, right? Pushing us to ask some tough questions.
Definitely. It forces us to ask. As we keep finding these shared molecular patterns between us and the microbes we encounter, how do we get smarter? How do we design interventions, vaccines, therapies that are incredibly precise?
Strong enough to fight the bad guys.
But discerning enough to never mistake friend for foe. What new ways can we find to tune our immune response, to get optimal protection without triggering those dangerous unintended consequences, that horror autotoxicus?
That really is the fundamental challenge, isn't it? Fine-tuning our defenses without causing friendly fire.
It's a major frontier for biomedical science moving forward, finding that perfect balance.